In 2025, geneticists extracted DNA from bones that had been sitting in a Colombian museum for decades. The remains came from an archaeological site called Checua, just north of Bogota, not in the city itself, not on the valley floor, but on top of an isolated hill rising from the Checua River Valley at 2,582 meters elevation. When Kim-Louise Krettek and her team at the University of Tübingen analyzed the DNA from the teeth and bones of these ancient people, they found something that should not have been there, a discovery. A genetic signature that appeared nowhere else in the Americas, not in ancient North America, not in later South American populations, and not in any modern indigenous group ever tested. These people existed 6,000 years ago, and then they vanished completely without leaving a single genetic descendant. The DNA told a story archaeology never could. Whoever these people were, they represented a lineage so isolated, so distinct, they took an entire branch of the human family tree with them. No trace of their genes survived in anyone alive today. This is the mystery of Colombia's ghost population. The answers buried in their DNA reveal how fragile human survival really was in the ancient Americas. The hill itself sits in the municipality of Nemocón, 7 kilometers north of the town center. The area is surrounded by the Checua River Valley. The landscape once supported dry forest. Plants included Dodonaea viscosa, Baccharis species, and Lupina species. It was a marginal environment for agriculture, but rich in one critical resource: salt. The Checua Valley sits in the middle of a halite-rich zone. Salt mines at Nemocón, Zipaquira, and Tausa surround the site. For thousands of years, this area produced one of the most valuable commodities in pre-Columbian Colombia. Salt meant preservation, trade, wealth, and power. The Muisca civilization that arose here centuries later built their economy on salt production and trade. They created specialized ceramic vessels called gachas, specifically for extracting salt from underground brines, heating them until the water evaporated and only salt crystals remained. This ceramic-based salt industry made the Altiplano wealthy. But that was millennia after the people buried on this hill had disappeared. Colombian archaeologist Ana Maria Groot first excavated the site in 1991. She returned in 1992 and published her findings that same year. The excavations revealed a cultural sequence spanning from 8,500 to 3,000 years before present. She identified stone tools by the thousands, more than 1,750 complete tools, scrapers, perforators, knives, over 2,800 stone flakes, the waste debris from the tool manufacture, all made from chert. Bone implements appeared throughout the sequence. Tools for sewing, for scraping hides, for working wood, similar to bone tools found at other highland sites like Aguazuque and Tequendama, 2 nearby rock shelters that were also occupied by hunter-gatherers during the same period. And in stratigraphic Unit 5B, at 60 centimeters depth, something extraordinary, a bone flute, a musical instrument carved from animal bone, dated to 7,800 years before present. Someone made music on this hill 7,800 years ago. Groot's excavations also uncovered human remains. Deliberate burials, not scattered bones from accidental deaths. People had chosen this hill as a cemetery. Radiocarbon dates from charcoal established a chronology: 8,200 plus or minus 110 years before present from the upper section of Unit 4, and 7,800 plus or minus 160 years before present from Unit 5B. The hill had been occupied, abandoned, and reoccupied over and over across millennia. Before we continue, where are you watching from and what time is it for you? What is one word for your mood while you are watching this video? Leave a comment with your answers. I love to see how far lost evidence reaches across the globe. If you are fascinated by how entire populations can vanish without a trace, hit like. It helps bring these stories to more people who want to know the truth about the first Americans. But the bones sat in storage at the Universidad Nacional de Colombia for decades. Ancient DNA technology was not advanced enough to extract genomes from remains that old in tropical conditions. Heat and humidity destroy DNA. Colombia's climate should have degraded these samples beyond recovery. Then, in the early 2020s, paleogenetics advanced enough to attempt the impossible. Cosimo Posth, a paleogeneticist at the University of Tübingen in Germany, partnered with Andrea Casas-Vargas at the Universidad Nacional de Colombia. Their goal was to extract whole genomes from bones that had spent thousands of years buried in the Colombian highlands, not just from Checua, but from multiple sites across the Altiplano. They wanted to build a genetic timeline, a genomic transect spanning 6,000 years of human occupation. They focused on 5 archaeological sites. Checua is the oldest. Laguna de la Herrera represents the Herrera period ceramic culture. Las Delicias is associated with early Muisca culture. Soacha sits near the end of the Muisca period, just before Spanish contact. And Purnia, in the Los Curos area to the north, is associated with guinea populations. There are 21 individuals in total, spanning from 6,000 to 520 years before present. The central challenge was DNA extraction. Ancient DNA slowly degrades over time. Double-strand breaks fragment the molecule. Cytosine deamination creates sequence errors. Microbial contamination can overwhelm the human signal. In tropical environments, degradation accelerates. Most ancient DNA studies focus on cold, dry settings, such as permafrost, caves, and high-altitude deserts, places where DNA survives. The team targeted the petrous bone, the hardest bone in the human skull, located in the inner ear. Its dense mineral structure preserves DNA better than any other skeletal element. They also extracted DNA from teeth, where the dental pulp can trap and preserve genetic material. The extraction process requires extreme contamination control. Modern human DNA is everywhere. A single skin cell from a researcher can contaminate a sample. The team worked in clean rooms, used sterile equipment, and followed protocols developed over decades of ancient DNA research. They created double-stranded DNA libraries, then used targeted enrichment to capture roughly 1.24 million single nucleotide polymorphisms across the genome. At the Checua site, the team successfully sequenced 7 individuals, the oldest in their entire study. All were from the pre-ceramic period, before pottery and before agriculture, when people still lived by hunting and gathering, 6,000 years before present. When the team compared the Checua genomes to a global database of ancient and modern DNA, the results made no sense. The Checua individuals did not cluster with any known population. The researchers used multiple analytical methods: principal components analysis, plotting genetic variation across 2 or 3 dimensions; admixture analysis, modeling each genome as a mixture of ancestral populations; F-statistics, testing specific hypotheses about population relationships; admixture graphs, building phylogenetic trees that allow for gene flow between branches. Every analysis told the same story. The Checua genomes were distinct. On multidimensional scaling plots of South American populations, they fell in a central position, suggesting an early divergence. They did not share excess genetic drift with ancient North Americans. They showed no special relationship with ceramic age populations from Venezuela, and they were not closely related to ancient Panamanians, despite Panama being the gateway between Central America and South America. Kim-Louise Krettek, the study's first author, described them as deriving from the earliest population that spread and differentiated across South America very rapidly. In other words, when humans first entered South America from North America, they did not move as a single unified group. They fragmented. Different bands split off, moved into different regions, and evolved in isolation from each other for thousands of years. The Checua people represented one of those isolated fragments, a founding population that settled the high plains around Bogota and stayed there. But the genetic data revealed something else, something concerning. This was not a large, thriving population. The genomes showed evidence of what geneticists call runs of homozygosity. These are long stretches of DNA where both copies of a chromosome carry identical sequences. This happens when both parents pass down chromosome segments that are identical by descent, meaning those segments were inherited from a recent common ancestor. In large populations with high genetic diversity, runs of homozygosity are rare and short. Recombination breaks up chromosome segments every generation, and mutations create new variants. People mate with individuals from diverse lineages. In contrast, in small, isolated populations where everyone is distantly related, runs of homozygosity become longer and more frequent. People inherit the same chromosome segments from both parents because those parents shared ancestors only a few generations back. The research team analyzed identity by descent across all 21 genomes. They used specialized software to detect these shared chromosome segments and to estimate how recently the common ancestors lived. The Checua individuals showed elevated identity by descent compared to later populations, and their genomes were distinctly homozygous. The study describes them as a comparatively small population of hunter-gatherers. There are no specific numbers. You cannot estimate population size precisely from 7 genomes. But the patterns were clear: limited genetic diversity, elevated homozygosity, the signatures of a small, isolated group. Mitochondrial DNA, inherited only through mothers, told part of the story. Haplogroup B2D appeared most frequently among the Checua individuals, followed by A2, then C1. Only one individual carried D1. 4 haplogroups in total across 7 individuals. This points to limited maternal lineages, consistent with a restricted founding population, or significant genetic drift over generations. The Y-chromosome, inherited only through fathers, told an even starker story. Every male in the Checua sample, and indeed every male across all 21 individuals from all 5 sites, belonged to Y-chromosome haplogroup Q, 1b, 1a, or to its subclades. That uniformity underscores extreme paternal continuity. Q1b1a is the dominant paternal lineage across Native American populations, but complete uniformity in a sample suggests either a very recent founding event or severe drift eliminating other lineages. This matters because genetic diversity is survival insurance. When a population maintains many genetic variants, some individuals will survive diseases, climate shifts, or resource collapses that kill others. Resistance alleles, metabolic variants, behavioral plasticity. When diversity erodes, a single catastrophic event can wipe out everyone because no one carries the genetic variants needed to survive the new selective pressure. Small populations face what geneticists call a mutational meltdown. Every generation, random chance removes some genetic variants. Beneficial mutations get lost before they spread. Harmful mutations can drift to high frequency before selection removes them. Inbreeding exposes deleterious recessives. Fitness declines. Population size shrinks further. Genetic diversity erodes faster. The spiral continues until extinction. The Checua genome suggested a population vulnerable to this process. Not yet in meltdown. Still surviving. Still reproducing. Still burying their dead on that hill. Generation after generation. But vulnerable. And then the researchers extracted DNA from later individuals buried on the same Altiplano. 9 people from sites associated with the Herrera period, dated around 2,000 years before present. 3 from the Muisca culture, around 1,200 years ago. 2 more from just before Spanish contact at Soacha, 520 years ago. None of them carried Checua DNA. No genetic continuity. The entire Checua lineage had been erased from the gene pool. Andrea Casas-Vargas, a co-author from the Universidad Nacional de Colombia, noted that this was unusual, especially in South America. In the Andes and the Southern Cone, genetic continuity is the norm. You see the same genetic signatures persisting through cultural changes, through the rise and fall of civilizations, through climate shifts. Modern indigenous populations in Peru, Chile, and Argentina carry genetic legacies traceable back thousands of years. Cultural change does not necessarily mean genetic replacement. But on the Altiplano, the genetic record showed a clean break. The hunter-gatherers vanished, and their replacements came from somewhere else entirely. The later Altiplano populations showed clear genetic affinities with ancient individuals from Panama and with modern Chibchan-speaking groups in Costa Rica and Panama. Principal components analysis placed the Herrera period individuals close to ancient Panamanians. F-statistics confirmed significant shared genetic drift. Admixture analysis showed they carried ancestry from a Central American source. The researchers concluded that around 2,000 years ago, a second wave of migration came down through the isthmus and completely replaced the Checua population on the Altiplano. These newcomers brought technologies the hunter-gatherers did not have. Ceramics. The archaeological record shows a sudden appearance of pottery on the Altiplano around 2,000 years before present. The oldest ceramic evidence found on the Altiplano dates to 2,500 years before present, about 500 BCE. One piece found near Tocorema has been dated to 2,750 years before present, making it among the earliest pottery in the region. This Herrera period, named after Lake Herrera, where early ceramic sites were first identified, is usually defined as ranging from 800 BCE to 800 CE, though some scholars date its beginning as early as 1500 BCE. Pottery changes everything. Not just storage and cooking. The Herrera people used ceramics to create an industrial economy based on salt. They manufactured specialized vessels for boiling underground brines, extracting the salt that made the Altiplano wealthy. These salt extraction ceramics allowed systematic exploitation of the halite deposits surrounding Checua. Trade networks expanded, population density increased, social complexity developed. Societies with ceramic technology for salt production had economic advantages. Societies without it could not match. The newcomers also brought language. Chibchan, language families, still spoken today in parts of Costa Rica, Panama, and Colombia, traced to this migration. The Muisca people, who built a complex civilization on the Altiplano centuries later, spoke a Chibchan language. That linguistic connection links them directly to the Central American migrants who replaced the Checua hunters 2,000 years ago. Branches of this language family are still spoken in Central America today, preserving the linguistic heritage of that ancient migration. But how? How does an entire population disappear without leaving a single genetic trace? This is where the science reaches its limits. The DNA can tell us what happened. It cannot tell us why. The researchers offer possibilities. Andrea Casas-Vargas mentioned climate conditions, disease, or lack of food, and she frames these as speculation rather than firm conclusions. Kim-Louise Krettek noted that the Checua may have migrated and lost their genes over time as they interbred with other populations. The study found no evidence of violence, no massacre, no mass graves showing trauma, no weapons embedded in bones. The Checua simply stopped appearing in the archaeological record around the same time the Herrera culture appeared. There are multiple scenarios. The Checua population could have been in decline when people from Central America arrived. Small populations are vulnerable to random demographic fluctuations. A few bad years, a disease outbreak, a drought. In large populations, some people survive these events and the population rebounds. In very small populations, everyone dies. Climate change could also have made the highlands uninhabitable for the Checua. The transition from the Middle to late Holocene saw environmental shifts across the Andes. Vegetation changed and rainfall patterns shifted. The dry forest that once surrounded Checua would have been sensitive to those changes. If the resources the hunters depended on disappeared, the population would have had to move or starve, or disease. The arrival of new populations brings new pathogens. People from Central America carried diseases the isolated Checua population had never encountered. No immune defenses, no resistance alleles. In epidemiology, virgin soil epidemics can kill 90% or more of a population with no prior exposure. The Checua, with their limited genetic diversity and elevated inbreeding, would have been especially vulnerable. Or the Checua could have integrated with the incoming population, but their genes were diluted out of existence. This is more common than people realize. If only a few Checua individuals survived and married into the much larger Central American population, their genetic contribution would shrink every generation. After 10 generations, one Checua lineage in a population 100 times larger would represent 1,000th of the gene pool. That would be below detection limits for sampling. We do not know which scenario happened. The true fate of the Checua remains unknown. We do not know if the Checua migrated elsewhere and their genes survive in populations we have not tested yet. Ancient DNA studies from Western Colombia, Ecuador, and Venezuela are still lacking. Kim-Louise Krettek noted that ancient DNA from those areas will be crucial in understanding how humans migrated into South America. The Checua lineage may exist in regions that have not been sampled. What we know is this. From roughly 9,500 to 5,100 years before present, people lived on that hill. Archaeological evidence shows 3 distinct chronological units. The first unit dates from 9,470 to 8,313 years before present, and it represents sporadic occupation. People visited the hill occasionally. They left stone tools, they hunted deer, they did not bury their dead there. This was a seasonal camp. It was a stopping point in their annual round of movement across the Altiplano. The second unit, from 7,580 to 6,280 years before present, marked a fundamental shift. Occupation became more intense. The frequency of archaeological materials increased dramatically. People started constructing residential structures, hearths multiplied, and crucially, they began burying their dead on the hill. The cemetery started. The bone flute appeared in this period. Graves were prepared with care, showing that this was when the hill transformed from a temporary camp into a place of deeper meaning, not just a hunting ground, but a home where ancestors rested. The third unit, from 5,892 to 5,190 years before present, showed full sedentism. People lived there permanently, or nearly so. The frequency of stone tools increased further. Implements for processing plants appeared, suggesting cultivation or intensive gathering. Evidence of domesticated guinea pigs increased. The Checua remains included both domesticated Cavia porcellus and a wild species called Cavia analimi. Ancient DNA analysis revealed that Colombia may have been an independent center of guinea pig domestication, separate from the better-known domestication in the central Andes of Peru. The presence of multiple Cavia species through time at Checua points to a complex, long-term interaction between people and these animals. Later, during the Herrera and Muisca periods, guinea pigs appeared in human burials, indicating a symbolic relationship had developed alongside their economic role as food animals. Population likely grew during this period, though it never became large. And then, around 5,190 to 5,052 years before present, the site was abandoned. People stopped coming to the hill. The cemetery stopped growing. The hearths went cold. For reasons we do not understand, the place that had been continuously or repeatedly occupied for over 4,000 years was left empty. What happened in the nearly 3,000 years between that abandonment and the appearance of the Herrera culture around 2,000 years ago, we do not know. No sites from this gap period, no remains, a blank in the record. And then Central Americans arrived with their pottery, their salt extraction technology, their Chibchan languages, and their different genes. The Altiplano was populated again, but by different people. The study, published in Science Advances on May 30th, 2025, represents the first ancient genomes ever published from Colombia. Cosimo Posth described it as filling a critical gap in our understanding of how South America was populated. Colombia sits at the geographic bottleneck where Central and South America meet, the Darien Gap, the gateway. Every human migration into the southern continent had to pass through this region. And yet until this study, we had no genetic data from ancient Colombia. We did not know who lived there. We did not know how populations changed. We did not know whether genetic continuity persisted or whether waves of replacement recaped the population. Now we know. At least on the Altiplano, replacement happened completely. Before they published, the research team did something most genetic studies do not do. They consulted with the Guardia Indígena Muisca, the modern indigenous community, descended from the Muisca culture that flourished on the Altiplano before Spanish conquest. Photographs from the study show the research team meeting with community members to discuss the findings and their presentation. Cosimo Posth explained that questions about history and origins touch upon a sensitive area of self-perception and identity for indigenous populations. Genetic disposition must not be viewed as equal to cultural identity. The Muisca people today carry a cultural legacy that connects them to their ancestors. Language is one part, though the Muisca language itself went extinct in the 18th century. Traditions, spiritual practices, knowledge of the land, and resistance through colonization and displacement continue to link people to their past. That legacy endures. That cultural continuity exists regardless of genetic ancestry. Central American migrants who arrived 2,000 years ago became the ancestors of the Muisca. The Muisca are not defined by genetics, they are defined by the culture they maintained and continue to maintain. This is the tension at the heart of ancient DNA research. Genetics reveals population movements and replacements that oral histories may not preserve. But genetics cannot capture lived experience. It cannot tell us what people valued, what they believed, what they mourned, or what they celebrated. Checua hunter-gatherers lived on that hill for over 4,000 years. We know they hunted white-tailed deer. We know they raised domesticated guinea pigs, possibly as part of an independent Colombian domestication separate from the Andean tradition. We know they made stone tools from chert and bone tools for working hides. We know they carved a flute and made music. We know they buried their dead with care, creating a cemetery that persisted for millennia. We know they lived in a landscape rich with salt deposits that would later become enormously valuable. We know their population was small, isolated, and genetically distinct. We know they occupied the same hill for millennia, developing from seasonal visitors to permanent residents. But we do not know their stories. We do not know their songs beyond that single bone flute. We do not know what they called themselves. We do not know what they thought about the mountains surrounding them or the meaning they found in life and death. We do not know if they knew their lineage was ending or if their disappearance caught them by surprise. All we have is their DNA and their bones. And the stone tools they left behind, a molecular ghost, a genetic signature that persisted in bone for 6,000 years but vanished from living people 2,000 years ago. Science can resurrect these genetic ghosts. It can prove they existed. It can place them in time and space. It can measure their diversity, infer their population size, and estimate when they disappeared. But it cannot make them speak. It cannot tell us if they saw their end coming. It cannot tell us if any escaped and carried their memories elsewhere. The gap between genetic confirmation and historical understanding remains vast. We can prove the Checua existed. We cannot recreate their experience. And perhaps that is the truest thing we can say about the deep human past. Most of it is gone forever. If you enjoyed discovering why Colombia's first people left only their DNA behind, watch the video on screen for more lost evidence.